This is part 2 of Chapter 6, Cast of Characters. This video will cover our naming system, binomial nomenclature we use to name all cellular organisms with a unique name. And in addition, we'll start looking at some specific examples of acellular microbes, the prions and the viruses. So again, for all cellular organisms, we provide unique scientific names. And the unique scientific name, it has two parts, two names, and hence we refer to it as binomial nomenclature, a two-name naming system. We want to remember that acellular microbes, such as prions and our viruses, won't follow the same system. So the two names, the two parts of the unique scientific name, the first part is called the genus, and we always capitalize the genus name, and we always want to make the genus name appear different from surrounding text. So if I'm just writing, I could underline it. If I'm using a word processor, I could italicize it. And the second part of this unique scientific name is called the specific epithet, or often it's referred to as a species name. It will be in lowercase, and again, we want to make it different from surrounding text, so we can either underline it or italicize it. A couple of examples of unique scientific names is the name for humans. We are Homo sapiens. So you can see here the genus name has been capitalized. The species or specific epithet is lowercase. We've underlined it to indicate it's a scientific name. And once we introduce a genus name, then we can use initial a shorthand for the scientific name. So Homo sapiens we could also write as H period sapiens. One of the bacteria we'll be looking at is Bacillus anthracis, or B. anthracis. This is the bacterium that causes the infectious disease called anthrax. Okay, so we're next going to move up. This is a great kind of concept map that one of your colleagues a few years ago put together, the microbial cast of characters. And you can see it's organized somewhat similar to your cast of characters handout. So we're first going to look at the acellular microbes, and remember, we know they're acellular because they can't synthesize a cell membrane. Okay, so if we ask, can they synthesize cell membrane, the answer is no, we know they're acellular. And our two examples are the prions. Prions are fascinating. Prions are abnormally folded, misfolded proteins that cells make. So they're not cells, but we would call them cellular proteins. And we'll discuss how we all have good normal cellular prions, but then when they misfold into this conformation, then they can stack, forming long filaments, and eventually can cause neurons to die. So the prions, which stands for proteinaceous infectious particles, are responsible for a group of diseases called TSEs, for transmissible spongiform encephalopathy. Transmissible means the disease-causing prions can be transferred from an infected host to a new host. Spongiform means something has the appearance of a sponge. And we can see that, sadly, the structure that's going to appear sponge-like with lots of holes in it is going to be the brain. So encephalopathy refers to a brain disorder. So this is a photomicrograph, thin section through the brain of an animal that died of a prion disease. Cellular material should be staining light pink, purple. The nuclei are staining a dark purple. But you can see that there appears to be holes in this thin section of the brain, and those holes are truly holes. This is what makes the brain appear like a sponge, having all these holes in it. And certainly with enough neuronal cell death, then the animal, the organism, is going to die. So the TSEs are 100% fatal. There's no treatment for them. Some examples are mad cow disease, Kuru, Creutzfeldt-Jacobs disease, which we'll be discussing in lecture. Now remember, prions are abnormally folded proteins. They lack DNA, they lack RNA, they don't have any genetic information. The misfolded prions are one of the most resistant structures known. Normal autoclaving will not destroy them, and this is a huge concern in hospitals. We'll go to our second acellular organism, and this is going to be a virus. Viruses can't synthesize cell membranes, so they are acellular. Viruses have protein, but in addition, they have genetic information, either DNA or RNA. Now, some viruses, when they replicate inside a host cell, when they escape, they'll take part of the host cell membrane. That will become the outermost layer or surface. And in those viruses that steal host cell membrane, that outermost layer is called the envelope. So we'll see that some viruses are what we call naked viruses in that they lack an envelope. Other viruses are enveloped. They have an envelope. The significance is that naked viruses can remain infectious in the environment for long periods of time. In contrast, envelope viruses, anything that damages the envelope makes it impossible for the virus to infect a new host, new cells. So luckily, envelope viruses are rapidly inactivated in the environment. The example we have here in this diagram is the enveloped RNA virus, influenza virus. And we'll discover in lecture that these RNA viruses, when their RNA is copied, lots of mistakes are made. So those mistakes are actually mutations. So RNA viruses like influenza virus can rapidly mutate, and we call that phenomenon antigenic drift. But in addition, we can have different strains of influenza virus infecting the same host, even the same cell. And when the influenza viruses RNA segments are being replicated and then they get packaged into new viruses to be released, we can get brand new combinations of influenza virus RNA in these newly replicated viruses. And that means the newly replicated viruses can be really genetically diverse. We think this process of reassortment, referred to as antigenic shift, is responsible for worldwide epidemics, pandemics. Okay, so now we're going to stop here, and the last video will be on cellular microbes and organisms.